Hi everyone. We're going to be picking up where we left off on cellular reproduction. So remember we ended kind of on cytokinesis, which is the division of the cytoplasm of the two cells. This is happening kind of overlapping with telophase, which is the division of the nuclei. So cytokinesis happens differently in animal cells versus plant cells due to the structural difference between these two cells. In animal cells, it creates something called like a cleavage, which is kind of like an indentation of the cell, and it creates the specific word for it is cleavage furrow. That's the word for this kind of like indentation where as the cell kind of closes in on itself to pinch off and then separate. And actually, if you looked kind of in a microscope and the cleavage furrow and what's causing the cleavage furrow, there's a contractile ring made of actin microfilaments that kind of, it's like a ring that starts to form around the cell membrane and start to pinch in and then finally closes off and separates the two cells. For plant cells, because it has a cell wall, it can't just pinch in like that. So what happens is it forms a cell plate. A cell plate is made up of these little vesicles that line the middle of the parent plant cell, and the vesicles contain some of the cell wall material. So it starts to form this like temporary cell wall in the middle of the parent cell, and eventually that cell plate fuses with the top and the bottom of the plasma membrane and the cell wall of the parent cell, and then it separates us two plant cells. Here's just a picture. You can see this is the cleavage furrow of the animal cell, so as it's pinching in, and this picture right here indicates the contractile ring of microfilaments that starts to close off and separate the two cells. Here in this plant cell, you can see the difference here where there's a cell plate, and the cell plate starts to line up and get stronger and eventually fuse with the top and the bottom. Okay, so after we finished discussing cellular reproduction, just the cycle it goes through, it's very important that the body is able to control the timing of cell division. You don't want all of your cells to divide whenever they want to spontaneously, and that's actually what leads to cancer. So the body has a very good way of controlling how cells divide. There are a couple different factors that influence cell division. The first one is anchorage dependence, this bolded term. And so what this means is that cells must be in contact with a solid surface to continue to divide. So if there's a free cell that's just floating around somewhere where it's not supposed to, it won't start to replicate. But you can see in this picture right here, if it comes into contact with a nice smooth surface, it's an indication to the cell that it's in a safe environment to start to divide. Something else that affects cell division is density-dependent inhibition. So this is the body's internal check to prevent the cells from getting too crowded. So once the cells start to touch each other, most cells will stop dividing when they form a single layer. So this picture shows that like if you have one single layer and you remove some of the cells will replicate just enough to reform that single layer, but it won't form more and won't like start to stack on top of itself. This picture right here, the cells stacking on each other, is just a representation of cancer cells, which don't have that check. The last factor that affects cell division is growth factors. These are kind of proteins that are secreted by the cells that let other cells know when to divide. So when you try to grow cells, for example, in like the laboratory, you need to put special growth factors in the medium that it's growing in for it to continue to grow. So here you can see a picture where you're just putting regular cells inside and nothing's happening to them because they don't have these growth factors that tell the cell to divide. Whereas if you put the growth factor here, then the cells start to divide very well. Another way that the body decides to whether or not to allow cells to divide, it has something called the cell cycle control system. The cell cycle control system represented here is just a network of proteins that regulate the different parts of the cell cycle. So there are certain stopping points in the cell cycle that let the cell know whether or not it should continue going forward or if it should stop dividing. These stop points are called checkpoints. And these are critical places where the cell can decide what it wants to do, whether or not it should continue going forward or if it should just stop in the cell cycle wherever it is and not continue forward. The most important checkpoint is G1 checkpoint. This happens during the G1 phase, which is just when the cell is growing, before DNA has been replicated. when the cell hits this checkpoint and it decides not to continue forward, it actually goes into something called the G0 phase. This is when it goes into the G0 phase, it's a permanently non-dividing state. So the cell will never go on to divide. So if it does pass the G1 checkpoint, it will go on to the other checkpoints. There's one at the G2 checkpoint, which is at the end of G2 phase. And then there's one called the M checkpoint, which is kind of in the middle of mitosis. So at each of these checkpoints, how the cell decides whether or not to go forward is by interacting with the environment. So when there are certain factors from the environment that indicate to the cell through a signal transduction pathway that can tell the cell whether or not it's okay for it to continue forward or if it should stop dividing. The book talks about cancer cells. This is kind of when cells go wrong and they don't have these natural checkpoints and they just continue to divide without any kind of check. Cancer in the United States kills 1/4, is responsible for 1/5 of the deaths. 1/5 of the deaths in the United States. And in China, it accounts for 20% of the world cancer deaths. So out of everyone who dies in the entire world due to cancer, 27% of them happen from China. So you can see it's a very serious concern. And these pictures right here are just pictures of cancer cells. As we said, cancer cells, they don't follow the normal signals that regulate the normal cell cycle. When you have unregulated cell growth, it can lead to something called a tumor, which is just a mass of these abnormal growing cells together. You can have two different types of tumors. One is called a benign tumor, and this is when the abnormal cells, they at least stay located in one spot. This picture right here is a picture of a couple benign tumors inside of the stomach, and these are better than the alternative because at least they're staying localized. But the malignant tumors are the ones you really don't want, and that's when the tumors can spread out to different areas. And this picture right here shows how all the dark spots represent cancer tumors, and you can see how it's been spreading all throughout this person's body. And metastasis is the name of the process in which cancer cells spread. So malignant tumors will go through metastasis and spread out to different parts of the body. There are a couple different ways to treat cancer. With benign tumors, you can just actually surgically remove them. And once you can just cut away that tumor, usually that's fine. But with malignant tumors, it's a little bit more tricky. You could do radiation, so subject your body in specific locations where the cancer is to high degrees of radiation, and that tends to kill the DNA, which kills the cells. Cancer cells actually are more susceptible to being damaged by radiation, so they'll die off more quickly, and they'll die off more quickly than regular human cells, the normal cells, but both would be affected. And then in chemotherapy, you actually give cancer patients drugs that will stop specific parts of the cell cycle to stop the cell from dividing. And this is why a lot of cells that go through a rapid division tend to be affected, like your hair cells. These cells, they kind of replicate very fast to produce like the proteins that can be used for your hair. And so when you get chemotherapy, it actually makes your hair fall out because those cells are also affected by the chemotherapy treatment. The problem with all of these kinds of treatments is that it's very harsh on your normal body. It's kind of like attacking the cancer cells, but it's attacking your normal cells. So they've actually they're trying to develop new personalized treatments just to make the cancer treatments a little bit more effective and individualized. And they do that based on the genetic profile of cancer patients. So when they diagnose the genes of the cancer patient, they can figure out like what specific genes are being attacked and see if they can find a pattern of which genes correlate with which drug treatments and make it a little bit more effective that way. All right, thanks so much for watching. Goodbye.